Hello students. Now let's continue with the Vibrio pathogen. Objectives: pathogenicity, pathogenesis, symptoms, laboratory diagnosis, epidemiology, prophylaxis, treatment. Now let's see pathogenicity. Before that, let me tell you, in pathogenicity, we always understand that there are some determinants. Now this is a fifth pathogen we are studying. Determinants of pathogenicity. Okay, maybe virulence factors or something. Everything is included in pathogenicity, so don't get confused. Now, virulence factors. Now I'll tell you some of the virulence factors and their biological effects. Cholera toxin. Its effect will be hypersecretion of electrolytes and water. Co-regulated pilus. Effect: adherence to mucosal cells. Accessory colonization factor. Effect: adenosine. Heme agglutination protease. Mucinase. Effect will be released bacteria from mucosal cells. Zona occludens toxin. Effect will be exotoxin. Accessory cholera enterotoxin. Effect will be exotoxin. Flagellum. Effect: motility. And siderophores. Effect will be ion sequestration. Okay. Several species of Vibrio are pathogens. We all know that. Now, Vibrio cholerae being motile bacterium, it's one of the major factors in the pathogenicity. Flagellar antibodies could prevent the invasion of the bacteria by immobilizing them, as well as somatic O and flagellar H antibodies can cause clumping and lead to arrest of motion of cells. Next, most disease-causing strains are associated with gastroenteritis but can also infect open wounds and can cause sepsis. Okay? They can be carried by numerous marine animals such as crabs or prawns and have been known to cause fatal infections in humans during exposure. Risk of clinical disease and death increases with certain factors such as uncontrolled diabetes, elevated iron levels, in disease like cirrhosis, sickle cell diseases, hemochromatosis, okay, and cancer or other immunocompromised states. Pathogenic Vibrio species include V. cholerae, the causative agent of cholera, V. parahemolyticus, and V. vulnificus. V. cholerae is generally transmitted by contaminated water. Understood? It can also cause foodborne illness, usually associated with eating undercooked seafood. And pathogenic features can be linked into quorum sensing, where bacteria are able to express their virulence factors via signaling. Okay. Symptoms: watery diarrhea, vomiting, thirst, dehydration, muscle cramps complications, muscular pain, renal failure, pulmonary edema, and cardiac arrhythmias. Next, pathogenesis. Source: ingestion of contaminated water, food, fruits, vegetables. Incubation period: it is 1 to 5 days. Now the causative bacterium V. cholerae enters the mouth with fecally contaminated food or drink. The bacteria attach to the epithelial cells of the small intestine. Then toxins enters the cell and prevents them from regulating secretion of water and electrolytes. Now the epithelial cells pump water and electrolytes from the blood into intestinal lumen, causing watery diarrhea. Understood what is going on? Now, shock and death occurs because of fluid loss from the circulatory system unless the fluid can be replaced. The bacteria exit the body with the feces. Okay. Now, laboratory diagnosis and treatment. Diagnosis: microscopic examination of stool is carried, generally non-productive because the organism is diluted in the large volume of watery diarrhea. We all know that. Now, culture should be performed early in course of disease with fresh stool specimens. Now, treatment, prevention and control part. Fluid and electrolyte replacements are crucial. Antibiotics reduce the bacterial burden and exotoxin production as well as duration of diarrhea. Doxycycline in adults, trimethoprim sulfamethoxazole children, or furazolidone in pregnant women is administered. Improved hygiene is critical for control, and combination inactivated whole cell and cholera toxin B subunit vaccines provide limited protection. Other vaccines are in development. Fine. Prophylaxis. It should be timely diagnosed. Hospitalization of patient, disinfection of the sources and identification should be carried out. Treatment of carriers should be done because carriers can be asymptomatic also sometimes. Disinfection of water, safeguarding water supplies from pollution, systematic and thorough cleaning of inhabited areas, fly control and protection of foodstuffs and water from flies. Next, washing of hands before meals and after using the toilet is necessary. Regular examination of personnel in food processing factories for identification of carriers is also extremely important. Understood? Now, if you have any doubts in this whole enteric pathogens session, maybe E. coli, Shigella, Salmonella, Campylobacter, Vibrio, you can ask me. Go through each and every pathogen first. See the lectures, understand. If you have problems, do ask me. Thank you.